Concept 5.3, Linked Genes. In this concept, we will be talking about how linkage affects inheritance and how genetic recombination affects inheritance. The discussion of X-linked genes in the previous concept has introduced you to the idea that there are specific locations on genes for each chromosome. Now let's discuss genes that are on the same chromosome and how that affects phenotypes. In fruit flies, the genes for body color and wing shape and size happen to be on the same chromosome. So those traits are inherited together. We would expect to see, since the genes are linked, a 1-to-1 ratio of parental phenotypes when a heterozygote is crossed with a recessive homozygote. If the genes are not linked, we would expect to see a 1-to-1 to 1-to-1 ratio, with 25% between the parental types and the recombinant types, or types that have characteristics that are different from the parental types, like this gray-bodied fly with vestigial wings, or this black-bodied fly with normal-sized wings. But what we tend to see is this. This is almost a 1-to-1 to 0-to-0, but it is 90% parental types and 10% recombinants. How did we get the recombinants if the genes are on the same chromosome? Remember synapsis and crossing over in meiosis? The homologous chromosomes here in the heterozygote have exchanged pieces. So the gray can end up on the chromosome with the vestigial wings, even though it didn't start there. This leads to greater diversity within the population, which is the point of sexual reproduction. We can tell how far apart the genes are on the chromosome by how frequently they recombine. If they are further apart, they are more likely to get split up during crossing over. If they are side to side, a break between them is less likely. We've been able to map genes on chromosomes using recombination frequency. Okay, that's it for this one. Make sure that you understand synapsis and crossing over in meiosis 1, prophase 1, specifically so that you can understand this idea of genetic recombination.